Bio-inspired materials and mechanisms involve the careful, systematic isolation of functional physical principles from complex, naturally occurring biological systems. Effective biomimetics relies on the rigorous translation of natural wave mechanics and energy capture into synthetic substrates. Consider the Shinkansen high-speed rail. Its aerodynamic profile isolates the fluid dynamics of a kingfisher's beak to eliminate sonic booms during tunnel exits. Competitive swimsuits utilize a similar extraction, applying the friction-reducing properties of shark skin to manipulate synthetic hydrodynamics. Nature has conducted millions of years of research and development on absolute physical constraints. It provides the blueprints. Engineers accelerate and specialize them. To understand this structural translation, we will conduct a direct comparative teardown of two specific systems: acoustic spatial mapping and photon energy transduction. These direct mappings are precisely how the discipline is tested in academic engineering, requiring a technical differentiation of biological and synthetic models. Mastering this module means looking at an organic structure and immediately extracting the mathematical architecture beneath. Biological echolocation evolved as a navigational solution for environments devoid of visible light. This diagram illustrates a split-screen architectural teardown of spatial mapping. Stripping away the animal tissue reveals a 5-step physical process of mapping three-dimensional space using acoustic wave reflection. Step 1 is sound emission. A biological or artificial source generates high-frequency acoustic signals, such as a vocal click or an electronic pulse. Step 2 is propagation. These sound waves travel outward through a physical medium, moving at a speed strictly dictated by the density and temperature of the air or water. Steps 3 and 4 are interaction and reception. The waves strike an obstacle, lose amplitude, scatter, and bounce a degraded echo back to specialized sensory receptors. Step 5 is interpretation. A central processing unit extracts temporal and spatial data from that returning echo to calculate distance and shape. Spatial mapping is entirely substrate independent. The exact same physics govern acoustic reflection, whether that wave is bouncing off organic tissue or a titanium hull. Engineers implement this exact 5-step reflection process in both clinical and naval environments. This schematic of an ultrasound transducer details how the biological vocalization is replaced by active hardware, emitting sound waves tightly calibrated between 2 and 18 MHz. The animal's sensory receptors, like a bat's ear or dolphin's melon, are replaced by synthetic receivers. Because human organs have varying acoustic properties, their density and stiffness alter the returning signal strength to build an internal map. This physics yields specific clinical advantages, providing real-time internal imaging completely free from the risks of ionizing radiation. But the hardware carries physical limitations. Medical ultrasound has constrained penetration depth, relies heavily on operator skill, and suffers severe signal occlusion if the wave hits bones or gas pockets. In naval environments, the physical medium shifts to water, utilizing sound navigation and ranging, or sonar. This formula overlay demonstrates the core sonar calculation. The range to a target is determined by taking the speed of sound in water multiplied by the time it takes the echo to return. Sonar is also bounded by its medium. Signal accuracy degrades heavily due to water visibility issues like sediment and algae, acoustic noise interference, and limited deep water penetration. Engineers intentionally trade the general adaptability of animal echolocation to construct highly specialized diagnostic and naval tools bounded by strict physical limits. We see a similar translation when pivoting from acoustic wave reflection to the engineering of energy capture, starting with the biological baseline, photosynthesis. The entire process hinges on a single high-impact quantum event, a photon violently striking a molecular lattice and ejecting an excited electron. This cellular cross-section highlights the light-dependent reaction. Solar radiation is absorbed by chlorophyll pigments housed within the chloroplast's thylakoid membranes. That photon's energy drives photolysis. It forces a water molecule to break apart, releasing free electrons, protons, and ejecting oxygen as a byproduct. The excited electrons are immediately captured. NADP+ is converted to NADPH, and ADP undergoes phosphorylation to become ATP. The system then moves into the light-independent reactions, the Calvin cycle, which take place in the stroma and do not require active solar input. During carbon fixation, atmospheric CO2 is converted into an unstable 6-carbon compound. This breaks down into PGA, and the previously generated ATP and NADPH reduce it into G3P. These G3P molecules are finally synthesized into stable carbohydrates, primarily glucose. The biological objective is the synthesis of stable, long-term chemical energy storage rather than the generation of a raw electrical current. Photovoltaic, or PV cells, are the direct engineered equivalent to the biological chloroplast. Capturing solar radiation and converting it to a usable grid has been a primary engineering challenge for decades. This split-screen comparison charts the divergence. PV cells share the baseline physics of using photons to excite electrons, but bypass chemical synthesis to output immediate electrical current. Modern iterations refine this capture. Crystalline perovskite cells and flexible thin-film layers maximize the structural efficiency of the photon trap. Multi-junction photovoltaic cells push this further by stacking multiple material layers, each physically optimized to capture a distinct wavelength of the light spectrum. This divergence reveals a critical engineering trade-off. Traditional PV cells generate highly usable, immediate power, but they lack nature's built-in mechanism for long-term energy storage. To solve the storage limitation of traditional solar panels, advanced engineering developed the bionic leaf. This system completes the cycle by mimicking the full photosynthetic loop. Instead of simply routing electrical current to a grid, the bionic leaf uses that captured electricity to forcibly split water molecules, mirroring biological photolysis. This artificial photolysis yields a tangible physical output, the generation of pure hydrogen gas and other synthetic biofuels. By combining biological chemical storage with engineered solar capture, the bionic leaf produces a scalable, storable fuel. Mapping these systems onto a single matrix aligns the two core physical domains covered throughout this analysis. This matrix charts the two physical domains we've covered: acoustic wave reflection and photon-electron transduction. For exams, echolocation maps directly to ultrasound and sonar. The biological vocalization equates to the active transducer, and the animal's sensory receptors map directly to the synthetic hardware receiver. For energy capture, photosynthesis maps to photovoltaic cells and the bionic leaf. You must differentiate the stored chemical output of glucose from the immediate electrical output of a silicon panel. Human engineering does not invent new physics. It refines, scales, and bypasses the evolutionary constraints of principles already proven by biology. Mastering bio-inspired engineering requires seeing past the messiness of organic tissue to recognize the universal wave and energy mechanics operating beneath.